Well, hello. Uh, I hope everybody's having a good day. And today we are having a live interview with Dr. Jeffrey Tompkins here. And we're going to be talking about the chimp human similarity, but we're also going to be discussing the new paper that has been published this year concerning the differences between chimps and humans. And I've got an intro here. I'm going to read this out. This is from the ICR website. Dr. Tompkins earned a master's degree in genetics in 1990 in the University of Idaho, where he performed. Sorry, let me get my glasses on. I should have had my glasses on. Uh, plants that actually I think that's plant science in 1990 in the University of Idaho, where he performed research in uh, hormones. He received his PhD in genetics from Clemson University in 1996. While at Clemson, he worked as a research technician in a plant breeding slash genetics program with a research focus in the area of quantitative and physiological genetics in soybean. After receiving his PhD, he worked at a genetics institute and became a faculty member in the Department of Genetics and Biochemistry at Clemson. He has become a Christian as an undergraduate at Washington State University in 1982, with a goal to eventually work as a scientist and author in the creation science field. In 2009, Dr. Tompkins joined the Institute for Creation Research. How you doing today, Dr. Tompkins?
I'm doing great. Actually, it wasn't my goal in 1982 to be doing this. That was God's goal. So
Right, right, right.
I, I,
I
I
was just
actually have a
reading off
test.
what you had at ICR.
Yeah, yeah, I didn't write that. Someone else did, but so we probably ought to get
Right.
that changed. But I actually have a testimony video up on YouTube that was just put up there recently, where I kind of give my life story and how I got into this, and it kind of goes way back to when I first got saved in 1982. So, anyways, but that's not why we're
Awesome.
here. So.
Right, right, right. Um, but of course, you've been on this channel once before, and we were talking about chimp human percent similarity and your particular studies that you've done. You've done five peer reviewed papers in Answers Research Journal concerning the chimp human percent similarity. I am curious though, are you going to go back to that because of these differences, these new studies that have come out? I know there's a new study on Y chromosome. And of course, this study this year that we're going to be talking about concerning concerning the differences between humans and chimps. Are you going to come back and write another paper concerning all those, given all this updated information?
Well, there's not much more that can be done. This lab in Seattle has produced telomere to telomere, end to end genome sequences for all the great apes. And so, the data is pretty much out. They had a similar project in 2018 that they did as well. And these are genomes that were assembled based on their own merits. They were not assembled onto the human genome as a scaffold. And so there's not a whole lot more that can be done at this point. I'll have to think about that, but I am hoping to put out another paper on the chromosome fusion, because I've done some Sentiny research around the alleged fusion site, and it's not supporting the evolutionary story, just like all the other data I published on that. So hoping to come out with something there, maybe in the next year or so. But
Right.
if you want,
Uh,
I can kind of go over the history of my work in this area and how it's kind of led up to where we are now, and
yes, go ahead.
how it even matches up. So when I first got to ICR in 2009, the president at that time, or actually the CEO at that time, asked me and another scientist who was a biologist to investigate the human chimp issue. And he said, we'll go to churches and people are saying, are humans and chimps 98% to 99% similar? This is what we're hearing. And so he asked me and this other scientist, he said, can you guys look into this? And he said, I want an answer in 6 months. And so I had just got there at the time. I didn't know what to say, but the other biologist said, the guy that was talking to us, his name was Henry. He said, Henry, that's going to take more than 6 months. So, anyways, he ended up leaving, and so the project kind of ended up on me. And so I thought, well, the first thing I need to do is just see what's been published, because whenever you get into a research project, you basically pour through the literature and see what's been published and where things are at. So there were 6 top papers claiming a 98.5 DNA similarity with humans. And so I looked at all these papers, and some of them I couldn't figure out where the data they threw out even went. Because the one thing I could tell, there was a lot of cherry picking going on. They were just comparing regions of human and chimp that were known to be similar. They were throwing out data that was not similar. And there were a number of papers where I could actually kind of factor back in the data that was being thrown out. And actually one of those papers was the first draft of the chimp genome. And based on the paper, I came up with anywhere between a 66 and about a, I can't even remember now, about 86% similarity, I think. It just varied. So obviously there was a lot of monkey business going on there with that kind of data. And so I'm thinking, and of course it wasn't 98.5. And so I'm thinking, well, I just need to start downloading my own DNA sequence data and working with that. So at the time, I began downloading what's called Sanger style DNA sequence. These are DNA sequence reads that are roughly about 1,000 to 1,200 bases long or so. And of course, once you trim up all of the sequence at the ends, it's low quality. You end up with something about 700 to 800 bases long. So anyways, I began downloading all these DNA sequences, and I used an algorithm called BLASTN, and I began comparing them to human. And I published an original paper, and I came up with a similarity of 70% in that paper. And I thought, well, that seems to match what Richard Buggs was saying at the time. And then I got an email from an evolutionist, and he said, I think he was trying to repeat my study, I think. And he said, I think there's something wrong with your algorithm. I think it's kicking out DNA sequences. And so apparently I was using the algorithm, in a high throughput purpose that it was never intended to be used for. I mean, I was literally blasting millions of sequences and in these monster data sets. And apparently the algorithm was not originally designed to handle that kind of input. So anyways, so I actually complained to NCBI. It's a division of the National Institutes of Health. And they have a bioinformatics group there. And I said, hey, I think this algorithm's messing up. It's kicking out DNA sequence. And so I shared my data sets with them. I shared my output with them. And believe it or not, they actually fixed the algorithm. So here's this crazy creationist, right, that's working with the government to get this algorithm fixed. So anyways, so they actually fixed the algorithm to where it would not kick out DNA sequence anymore. And then I published a number of papers after that. And I actually published a very interesting paper in 2016 where I actually looked at all of the DNA data sets. I think there was 101 chimpanzee DNA sequence data sets that span from about 2004 to I think about 2010. And the nice thing about these Sanger style DNA sequencing data sets is that they have an XML file attached to the DNA sequence that has all this metadata in it. And so I began putting all this metadata into a relational database. And so basically the metadata tells you a lot of different things, but most importantly, it tells you when and where that DNA sequence was produced. Surprisingly, a fair number of the data sets didn't even have any metadata, which was really strange because you basically want that metadata so you can go back and troubleshoot, DNA sequencing projects with Haywire or whatever. So I thought that was a little weird. I ran a genome center for, a number of years. That metadata can be pretty important for troubleshooting stuff. But anyways, Be that as it may, I discovered that the first half of the Chimp Genome Project, which actually went into the publication of the first draft of the Chimp Genome in 2005, that data set was 5 to 6% more similar to human than the later data set was. Now that's a pretty sizable chunk, 5%. And I also found out that the DNA sequences were hitting with a much higher identity of around 90%, and compared to 85% for the later identity for the later data sets. And so basically, it's my surmising at this point that was human DNA contamination in those early data sets, those early chimpanzee data sets, because there were a couple of papers that had been published about that same time that I was working with these data sets, where they showed significant amounts of human DNA contamination in non-primate data sets, from everything from bacteria to zebrafish. In fact, for zebrafish, I think 3 million bases on zebrafish chromosome number 3 was entirely human. And this is because human DNA gets into the reactions and ends up being sequenced. So, and then subsequently ends up being assembled into the genomes that you're sequencing if your bioinformatics people aren't paying attention for that. So anyways, since then the technology has changed where the DNA contamination issue is not that much of a problem anymore, because now we have all of this long read DNA sequencing where you can pretty much figure out right off the bat, if your 30 to 50,000 base DNA sequences is your target sequence or contamination. So anyways, I think we've gotten past that now, but I think that's interesting because not only you have these chimpanzee data sets at the time, having a considerable amount of human DNA in it, maybe even up to 5%, but they're assembling them onto the human genome on top of that. So you've got these incredibly humanized chimpanzee and ape genomes well before the era of all of this long read DNA sequencing that we now have. So anyways, in 2018, there's a science paper that was published by that genome group up in Seattle that published this recent paper we're going to be talking about. And that's headed up by Evan Eichler. He's a real famous genomics guy known around the world. And he's got a huge lab up there at the University of Washington in Seattle. And in 2018, they actually used this long read sequencing and resequenced the ape genomes, chimp, orangutan, gorilla. And they actually said in that paper that previous versions of the ape genomes were humanized. And it was just something I've been pointing out for years. So they actually said that at that time. And to my knowledge, when I read that paper, I couldn't see anywhere where they came out with a specific genome-wide DNA similarity. But anyways, they did make some startling revelations about previous genomes being humanized. And they claimed that their genome that they had produced was a de novo assembled genome sequence, meant that it was sequenced on its own merits. So based on the overlaps between these long reads and some other technologies, they were able to put the genome together without hanging it onto human as a scaffold. And so one little interesting excursion to that is that they had a link in the paper to where you could download these new genomes, these new ape genomes. And it was a link to a very famous genome browser website in the United States. And so I clicked on it, and it was a dead link. Now this is a science paper, mind you. Why would it have a dead link in it? This is a top tier journal, right? So I wait a few more months, click on the link, it's a dead link. And I can't remember how long I waited, but it was a long time. And it was still a dead link. And so I contacted this lab that was processing these DNA sequences from Seattle, doing the bioinformatics and trying to get it up in a genome browser. And I said, what's up with this ape sequence that should have been up by now? You had a link to this paper that was published. And So they said, oh, we're basically, this is a paraphrase, but we're basically fixing the data. I'm like, okay, well, that doesn't sound good. But anyways, so anyways, the DNA sequences that were produced in that 2018 project, I downloaded a random number of those from the public database at NCBI. And on average, those sequences, I downloaded 18,000 random sequences. On average, they were each 30,000 bases long. And so when I blasted those onto human, and now I used the most liberal gapping parameters I could, so it could get through the gaps, the maximum gap size that this algorithm could handle. On average, I could only assemble 10,000 of those 30,000 bases on average for those DNA sequences onto human. So there's 20,000 bases where I couldn't even assemble onto human because the algorithm broke off because the DNA sequence became so dissimilar. So based on those 10,000 bases that I could assemble onto chimpanzee, I got an 84% on average DNA identity. So that's very interesting. We'll talk about why that is a little bit. And as you know, this past year, Robert Carter, who was another creationist, began looking at that data that I had, and I actually put it up in GitHub so anybody could look at it. And so Carter downloaded it, and he said, you've got two peaks here in your data, and that one of these peaks looks like hits that are really super similar to chimp, and the other peak looks like a lot of reads that are very dissimilar to chimpanzee. And as I was actually reading through my papers, I had a 2016 paper where I actually discovered that myself and forgot about it. I just read the paper today, and it's like, whoa! I had already talked about that before Robert Carter even noticed it. So in that 2016 paper, I did find two different data sets. One data set was very similar to chimp, and the other was very dissimilar. So apparently, we have parts of our genome that are very similar to chimp, probably because of these common proteins that most mammals share. Anyways, and then we have these regions that are very dissimilar to chimp, which are human, these human unique regions. So, anyways, having said all that, earlier this year, I think it was in April, there was a paper published in Nature. So it was the same group run by Evan Eichler up in Seattle at the University of Washington, and they basically sequenced, had another sequencing project in end-to-end. They call it telomere to telomere sequence. So it was even more complete than their 2000 and extensive than their 2018 paper. So anyways, the paper itself was about 17 pages, and it had a really strong evolutionary bent to it. I don't know if that was because they're trying to make up for the last paper that where they were kind of really being candid about what the data really said. But it was really hard to figure out from that initial 17-page paper what was really going on with the overall DNA similarity, because it was kind of I don't know how to put it, but anyways, it wasn't clear. But they did have a supplemental material section that they put up on the web, and that was 171 pages long. I mean, that is like a book. And so I figured, well, probably the real data that we're trying to find here is in this 171 pages. And sure enough, and I actually published a news article on this publication, and I've got it right here. Maybe you could put it up for your viewers in the video later. But I talk about the data that's of most interest here are tables Roman numerals 3.119 and 3.20 in the supplementary information on pages 33 through 36. And so basically they gave the overall identities for chimp compared to human. So human was the target, and then chimp was the query. So based on that, they actually very interestingly, had two different types of genomes. They had a haploid genome equivalent. Now, humans, apes, most animals have diploid genomes. So you have one from your mother and one from your father. And if you were just looking at one of them, it would be called a haploid genome equivalent. If you were looking at both of one from your mother and your father, that would be a diploid. So anyways, they created a haploid genome, a consensus haploid genome sequence, and then they created a diploid genome sequence. So with the haploid, comparing it to human, they came up with 85% DNA identity compared to human. I think they had about 6% they couldn't even get to match up onto human. And I don't know if that was included in the previous number or not. But anyways, 85%. Well, that was very similar to a number of papers I published in 2015 and 2016. But what's interesting is that with the diploid genome, the chimp diploid genome they queried onto human, they came up with 84% similarity. Of course, querying the diploid onto a diploid human is considerably more accurate than just a haploid genome. So anyways, 84% DNA similarity, I think about 6% or so that could not even match up onto human. So that's the exact same number I had in my last paper in 2018. 84% DNA similarity between human and chimp genome-wide. And of course, they actually did a massive genome-wide comparison using super high-tech algorithms where you can actually do that. The BLAST-N algorithm is just a local alignment algorithm. But they use some high-tech algorithms where they could actually compare entire genomes. So anyways, this is a massive lab with millions of dollars of funding. So I pretty much trust what they're putting out there, even though they're burying it in the 170-page supplemental material section. So anyways, so I don't know what else I can do from here as far as doing more research. Like I say, I've got a paper coming out where I've done some more research on the chromosome fusion subject. I'm thinking about doing some more papers on some of these pseudogenes that I've looked at in the past and kind of beefing up those with this new data. Because I'm finding out that this new telomere to telomere data is much better than what we've had in the past. And so it's much more accurate. And so I'm just going to keep plugging along, doing what I can do from my end. So anyways, I basically have been vindicated on this, as not only myself, but Richard Buggs as well. He got in a big discussion back and forth with a bunch of evolutionists on the BioLogos website, which is a website for Christians claiming to be evolutionists or theistic evolutionists. And basically, they're just evolutionists. They buy the whole neo-Darwinian thing, hook, line, and sinker. And so Richard Buggs was going back and forth with these guys. And of course, Richard Buggs is he's a professor in England at the University of London, Queen Mary. His specialty is actually genomics and evolution, so he knows what he's talking about. And so he was looking at all the different versions of the chimp genome, and I think the last one he looked at was Pantros 6. And he said basically Tomkins is right. That's about 84% similar, based on all the last Z alignments he downloaded from the whatever genome browser he used. So anyways, so both myself and Richard Buggs have been saying, years ago, 84%. And now this massive study comes out and it's 84% maximum similarity. And that doesn't include what is present in human, absent in chimp, what is present in chimp, and absent in human. How can you compare that? You can't, because there's nothing to match up against. So anyways, so you still have to factor that in. How we do that at some point, I don't know. But anyways, 84% maximum is probably much less than that if you were able to factor in what I just mentioned. So I think it's safe to say that the human and chimp genomes are radically different at this point. Some of them might say, well, 84%, maybe that's enough for evolution. But all of these theoretical models of evolution based on what they call a neutral model, basically they require, if you're going to get a common ancestor, say 8 to 6 million years ago, you need, basically your data needs to be 98.5 to 99% similar for your statistical evolutionary model to work. Now, these are not empirical models. And one thing that's really interesting is there was 2 papers published in 2012. One was in Science, the other was in Nature. And they actually looked at thousands of human exomes. So what's an exome? So an exome are all the exons of the protein coding genes in your genome, in a human genome. Now, these are protein coding exons, so these are highly intolerant of mutation. And so they found all these point mutations in thousands of humans in Europe and North America. And so what they did is they used empirical models on population growth, how fast populations grow, how many babies people have, and they just basically used empirical data. They didn't try and calibrate their models with deep evolutionary time. And so this one paper said, we believe, based on the mutations in these exons, that the human genome is no more than about 5,000 to 10,000 years old, and before that, it just flatlined. Well, that's ridiculous. And the other paper came up with about the same numbers. So what happened about 5,000 years ago? Well, the global flood, where you have this bottleneck of Noah and his 3 sons and their wives repopulating the earth. And so that completely lines up with Scripture. So when you use empirical data with genetic data, like with humans, you get biblical timelines. And actually, there was a study published in the mitochondrial genome back in the late 1990s. I think it was 1997, where the researcher looked at the point mutations in the mitochondrial genome, and he just used empirical data as well for his model, and he came up with the mitochondrial genome being about 6 to 7,000 years old. Well, that would go back to the time of creation when Eve was created, because you inherit the mitochondrial DNA from your mother. And so a lot of these studies that use empirical data to calibrate their genetic data come up with biblical timelines. And so I'm not sure how I got into that, but I got into that because these evolutionists, when they do these theoretical models and try and calculate, past population sizes and all this nonsense, they're all calibrated with deep time. And they're highly hypothetical, and they are not empirical, really at all. And so when we look at the empirical data, it totally supports the Scriptures. Humans were created about 6,000 years ago in the image of God. And they did not evolve from chimps. We have lineages in the Bible that go directly from Christ to Adam, which is 4,000 years. And then since the time of Christ, it's been 2,000 years. So we have a 6,000 year timeline back to Adam and Eve. And there's actually secular scientific studies that actually support that have been published.
Right. Thank you, Dr. Tompkins. I appreciate it. And I actually had a critic that had emailed me this morning. Your favorite evolutionist there, Glenn Williamson. He emailed me this morning and said that have Tompkins explain these papers, and he was talking about chromosome 2 fusion. And I'm like, well, we're not really doing chromosome 2 fusion this time.
I just gave
But
an in-depth hour-long talk on the chromosome fusion about a month ago on the BioLogos website, which I'm sure is up on YouTube. So
yeah, I've seen that.
you want to ask me something specific about chromosome fusion? Let's talk real data. Let's not, what
Yeah.
data point did I say there that didn't make sense or whatever?
No. It makes sense to me. Just a critic sent me something this morning asking me about that. I guess he's Berlin is always about chromosome two fusion. You could be talking about no telling what, and he'll be trying to get on that subject matter. But yes, I've seen your Biologos one. It was actually very good. Was it Logos or Biologos?
I'm sorry, Logos Research
Logos.
Associates. Yeah,
Yeah,
yeah, I messed that
yeah.
up.
Because
Yeah, Logos
Biologos is a
Research.
theistic evolutions.
Yeah, right. Yeah, they
Yeah.
would not have me on their site. Thanks for
No.
clarifying.
Right. Um, but in terms of the paper, though, you talked about how they tend to, and I've heard this before myself, that they seem to bury this information in the paper. I mean, it's there, but you have to actually dig into it, look at the methodology that they're using, go to the supplement information because a lot of the stuff is in the supplement information. Is that what you're referring to? The fact that they don't come right out and say it, but yet it's there. Is that what you're saying?
Right. Yeah.
Yes,
Yeah. I mean,
absolutely.
who's gonna spend their time look at 171 pages of boring data? I mean, so anyways, and I send emails to these labs with valid questions, and I never get a response. And so, I send them to multiple researchers on these papers because there's tons of them, and no one wants to respond. So, anyways, yeah, what can you do?
Um, right. And I also read the updated post by Richard Buggs as well. Some people thought Richard Buggs had changed his mind about the 84%. He actually hasn't. Um, he told me that in an email that he still holds to that position. But he also did an updated post since that information came out. He also came out with a similar thing with chimps and humans, no more than 85%. I've heard some people say that it depends on how you count differences. Um, as far
That's
as
that's
critics go,
yeah,
go
that's
ahead.
absolute nonsense. The differences are differences. And how do you deal with these regions, like I said, that are present in human, absent in chimp, that are unique to chimp and unique to human? Let's begin factoring those in. Of course, I don't think evolutionists really want to go there because that would make their case even worse.
Right. Um, and on that differences I think it's they're trying to justify cherry picking the data. Like, well, we don't really want to count the repetitive areas. We don't want to, they have this junk DNA argument that they made, which is another question I had because I've heard that some critics try to argue that these differences are found in the non-protein coding regions. How would you respond to that objection? Because I think the idea is that they're trying to claim that it's functionally irrelevant. So is what I think they're trying to argue. But I think it goes that down to the junk DNA argument.
Yeah, so basically you've got this entire ENCODE project going on in the United States, and you got other versions of it going on in Japan and Europe. And these people are actually, instead of pontificating about human evolution, they're actually trying to look for things that are underlying heart disease, cancer, diabetes, things like these. And they're trying to figure out what is all this information in the genome doing. And so, in the second draft of the Human Genome Project in 2004, that was when they said, okay, we need to figure out what all this DNA sequence means because we really don't. And so they started what's called the ENCODE Project, Encyclopedia for DNA Elements. And so it's a massive project because it has massive amounts of funding. It takes into account gene expression, 3D genome structure, and they're basically trying to figure out what the genome is doing. So in 2007, they published their first round of papers just looking at about, I think it was only 1% of the genome at that time. And they basically said that the human genome is highly functional at this point. One guy even published a paper shortly after that and said The human genome is an RNA machine. And that was Maddock in Australia. And so since then, they published a second round of papers in 2012 where they looked at the whole genome. And they found out that there was up to 3 times as many long non-coding RNA genes as there were protein coding genes that are highly functional. So now long non-coding RNAs are one of the hottest areas of research that are being funded by the NIH, because these long non-coding RNAs, or some of them are staying in the nucleus and doing things regulatory. Some of them are exported into the cytoplasm where they do a variety of different things. Some of them are even exported out of the cell in exosomes and transported to other tissues in the body where they do things. And so it turns out that long non-coding RNAs are literally the workhorses of the genome in many respects. And so, and they're basically encoded outside these protein coding genes in these so-called junk areas. And in 2012, the head over the bioinformatics for the ENCODE project, who was in England, he basically said at this point, 80% of the genome is biochemically functional and active. And he also said in 2012, that number is likely to go to 100%. So that was 2012. That was 13 years ago. And I haven't seen anything to the contrary since then. So basically, the human genome is literally transcribed from end to end. This nonsense that it's full of junk is ridiculous. And you don't have these biomedical guys saying that. You have these theoretical guys who really, there's not that many of them, because it's hard to get grant funding by just sitting there pontificating about evolution. If you want grant money, you're going to try and solve real problems like health problems in humans. And so tons of money is going into these big biomedical research programs where they're actually studying the human genome to try and figure out how they can cure and deal with a lot of different human diseases. So anyways, I want to go back and talk about those human exome papers that were published that I talked about that showed the human genome was only about 5,000 to 10,000 years old. Is what they were looking for was point mutations in those protein coding gene exons. Well, it turns out that over 80% of those point mutations were connected to human disease, like diabetes, heart disease, cancer, things like these. So in other words, when you get mutations, which are supposed to be the driving force of evolution, you don't get anything good, especially when it's going on in an important protein coding section of the genome. And so anyways, so that paper, it even highlighted that aspect of the neo-Darwinian paradigm. That's why you have so many evolutionists now who are biologists going over to what they call the third way of evolution. I don't know if you've heard about that or not, but they actually have a website called the thirdwayofelvolution.org. And these are all dissenting biologists that say, hey, the neo-Darwinian paradigm is totally bogus. Mutations are not a source of genetic novelty or genetic complexity. And we don't have a mechanism for this. Everything looks like it's highly designed. Everything operates in tightly, highly functional integrated networks, gene networks. Look, we need another model for evolution. And so they call themselves the third way of evolution. And they don't deny that some form of evolution has taken place. They just say, oh, we need more data. Well, how much more data do you need? I mean, that just shows you how when someone has a presupposition of they want to believe everything evolved from a single cell, billions of years ago, and there's no God, that's what they want to believe. That doesn't matter what you tell them, how much data you give them, they're going to hold to their guns because they want to reject God and be self-independent. So
Absolutely. And on the ENCODE project, they actually showed that 80% of our genome is functional. And you said by what date did they think that it could be pushed to 100%?
well, that was 2012 where they said 80%, and the guy that said that, I think his name was Ewan Burney, I think, in England. And when he said that, you would not believe the heat he took for saying that. And so I don't even know if he still works with ENCODE anymore, but he took tons of heat for what he said. And so, see, that's what happens. These evolutionists come out and say something bold like that, and they get so much kickback that they shut up. And I think that's what happened, with this brand new paper that came out in 2024. That's why they buried the non-evolutionary data in their 171-page supplemental material. So no one has said at this point the genome is now 100% functional, but they're sure funding tons of this research, and it's sure growing like wildfire. And so, there was an interesting study that was done. I'm trying to think of when it was done, maybe 2014, with this lady who was a cytogeneticist. I think she was up in Pennsylvania. She basically took RNase, which is a enzyme that destroys RNA, and she put it in with proteins. Or I'm not proteins, with chromatin. So chromatin basically is DNA wrapped around histones and coated with RNAs and other things. So when she put this RNase in with these chromosomes, and it destroyed the RNA, the chromosomes collapsed because all these RNAs were forming basically an infrastructure, a functional infrastructure coating the chromosomes. And so I thought that was a very interesting paper as well. But basically, the genome is transcribed from end to end. Every bit of it is doing something. And this whole idea that there's junk DNA out there, spitting out wonderful brand new genes randomly with intact promoters and intact, signals throughout the gene, intact introns and exons, and intact signals that tell it where to go after it's transcribed, and tell it how to be transcribed, and tell it how to be translated, and all this. If you think something that complex is going to get spit out of random DNA and magically integrate itself into a gene network with all of these signals and function perfectly, you're totally deluded. And so, yeah, the genome is highly complex, and it's more complex than we ever thought. And more papers come out every day just showing the incredible complexity of all these genomic systems that are going on. And of course, they're all integrated with one another, functioning as a precise, these cells function like just amazingly precise machines with thousands of networks, thousands of components. It's absolutely incredible.
Right. How does this fit with the Y chromosome research? I know that there was a paper published a few years ago that showed humans and chimps at 26% on the Y chromosome, and I know this paper also did some research on the Y chromosome as well. So how does this fit together with that earlier study on the complete sequence of the Y chromosome?
Well, the autosomes were 84% similar, chimp compared to human. The X chromosome, which is the female chromosome, one of those is shut down in females. And of course, males have an X and a Y, females have an X and an X, and one is shut down. So the X and the autosomes compared to human were about 84% on average, similar. So, or 84% DNA identity. The Y chromosome was less than 10% similar to human. And of course, this matches up well with that 2010 paper that was published on the chimp Y chromosome, where they actually had created a real physical map to hang based on chimp DNA sequence to hang the chimp DNA sequence reads on. And so anyways, yeah, and that paper, they basically said something like, the chimp chromosome was about as similar to human as chicken to a human or something like that. I mean, it was just, that paper was, I published some news articles on that, and yeah, it totally supported that. And of course, that was done, 15 years ago. So yeah,
Right.
the Y chromosome is really sticks out as a major non-evolutionary factor in this whole equation as well.
Right. Um, uh, the Y chromosome in that one paper a couple years back, it showed like 26% similar at the Y. So that kind of blew everything. That was like an exploding piece of information there. Um,
Yeah,
that
that didn't
humans,
include all the yeah, that didn't include all the repetitive stuff either. That was just focused on the gene rich areas.
yes, absolutely. Um, um, do you think there's a bit of an agenda there on the paper that they intentionally did not want to come out and admit that there is a uh, these differences actually make humans and chimps much less similar than what was originally thought. Do you think there was kind of like where they knew that where this data would lead, but they didn't want to come out and say that in this paper? You think there was kind of like an intent behind it?
I mean, I can't say for sure, but I would say probably. I mean, yeah, you
Uh,
know, if that's you're trying to toe the party line and you come up with data like that, it's embarrassing, right? So,
some people say that with um, the uh, I lost track of my train of thought. I was gonna ask a question. Um, do you see that the okay? I heard that what was the percent similarity between gorillas and humans in that paper published this year?
I don't remember. Yeah, I don't remember. It was less than human and chimp.
Right.
So,
Um,
yeah, actually, it's interesting. And of course, orangutans less than that. But it's interesting. There was a paper published in 2009 where they determined that based on anatomical traits, uh, the humans were most similar to orangutans. And and of course, this was when all the DNA sequence stuff was coming out, showing that wasn't the case. But as far as anatomical traits, we share more traits, anatomical traits with orangutan than we do chimps or gorillas. And actually, it's a hodgepodge. You know, there's some traits we share with chimps, and some we share with orangutans, and some with gorillas, and not chimps, and not so on. You know, it's just a mixed up deal, hodgepodge. So there's no clear evolutionary reasoning if you look at anatomy or DNA to link, humans with chimps or gorillas or orangutans. I think all these apes, chimps, gorillas, and orangutans are all their own created kinds because their DNA is actually quite different from each other as well. So yeah, humans are humans, chimps are chimps, orangutans are orangutans, and gorillas are gorillas.
right. Um, and I know that this paper was referenced by uh, the it's an Intelligent Design Institute. I'm trying to think of the name. Discovery Institute. I know
Discovery
that there was a
Institute,
guy
yeah.
on there, yeah, and named Luskin, and he basically presented this paper online. He's getting a lot of grief on that. And is it my understanding that the position that you should have and that everyone else should have really is to look at all of the differences that are there, because that's the one thing that a lot of people are doing, and this is something that you've noted early on and you've noted in your papers as well, that people were cherry picking the data, and it's almost like if they need that 96 to 99% so badly that they're willing to sit there and try to discount differences that ought to be counted as differences, just so that they can get that number. Is that Is that how you're looking at it? That's their gold standard. That's what they're aiming for, and so they are engaging in a kind of widespread cherry picking of the genome.
Well, it depends on who you're talking to. Is this some, you know, professional academic in a genome center that I'm talking to, or someone that's on YouTube just criticizing, and they don't have that background. So I don't know. Like I said before, people have got their presuppositions that they hold to, and doesn't matter what the data says. Um, but actually got some backwater on that Discovery Institute post. So that was first put up by Casey Luskin. And, of course, he was overwhelmed looking at this 171 page supplemental material, where you start with all that. And he found some graphs in there, and he was trying to, but based on, where the lines were in these graphs, he was trying to figure out what the similarity was. He missed that table. And so he put up that original article. And so I called him up on the phone. He's a friend of mine. I said, "Hey, Casey, the hard data you're looking for is in this table." And I said, go to these two tables on page so and so and table so and so's, and look, and their data's just right there. You don't have to try and figure it out from some graph. And so it's my understanding he published a second article based on that tip that I gave him. So anyway, so that's what happened
Awesome.
there. But yeah, so he jumped all over that when it came out too. So
Right, right, right. Um, and of course, evolutionists did do what I like to call damage control, where they have to post videos and try to figure out how to way to make this softer blow. Like I said, that they try to argue that it depends on how you count differences, and that they try to argue
just
that yeah that this is in the repetitive region and that sort of thing. Some people say that the repetitive region is rapidly evolving. Is just them presupposing that this region of the genome is uh, evolving fast because of how many differences exist in the repetitive regions? Because I know that the repetitive regions is usually where most of the differences are. So, how would you respond to someone who assumes that this is a rapidly evolving region of the genome?
well, first of all, they're not. They claim that it's rapidly evolving because the differences are tend to be more in those areas. But these areas are covered up with long non-coding RNA genes as well. And if the whole purpose of the product of that gene is just to stabilize a certain section of chromosome, it doesn't have to be, so tight that it can't absorb a few mutations compared to, say, an exon in a protein coding gene. That doesn't mean it's rapidly evolving and not doing anything. Yeah, these intergenic regions of the genome are hot areas for research, for looking for function, for, like I said, all these human diseases. They're trying to crack and figure out what's going on with these genetically. These intergenic areas are hotbeds for research. Your long non-coding RNA genes are in these areas. Your long intergenic non-coding RNA genes are in these areas. And yeah, it's this whole junk DNA thing that actually goes back, believe it or not, to the late 1960s, before DNA sequencing even became a thing. It was based on the neutral model of evolution they were creating at the time in the 1960s. And then that just spilled over into the DNA sequencing area, where when they sequenced the human genome in 2001, the first draft, they had all this DNA sequence. They didn't know what it did. They knew there's uh, over 20,000 protein coding genes in it, but that's about all they knew at the time. And so these ideas from the 1960s spilled over into that, and combined with this fact that they had no clue what the genome was doing in between all these protein coding genes, and those two things just kind of meshed together to form this idea, this modern idea of junk DNA, which has now been totally trashed by the ENCODE project. It's been put in the trash by ENCODE, junk DNA, where it belongs. So, and that was in 2012, where they came out with over 80% of the genome is biochemically and functionally active. Yeah, 13 years ago. Where do you think they're at now with all this? And so, yeah, so I see papers coming out incessantly on all these long non-coding RNAs and all these intergenic regions that are doing all kinds of important things. So, anyways.
Right. And these different, I think that if I'm not mistaken, Richard Buggs's method is to include all the differences in the. Is that right?
Well, he downloads these chained genome alignments using an algorithm called LAST-Z. That was actually a deprecated algorithm that, but apparently they've been maintaining it at the UCSC Genome Browser website to perform on modern machines. I don't think it's public, you know. I mean, I've used a version of it that would still work on modern computers in a paper I think I published 2015, I think. But anyways, but there's other, you know, good global alignment algorithms now where you can actually put entire genomes together. But you need a lot of computational power. And of course, that's what they've got up in Seattle there.
Right. All right. I guess I'm gonna open up to some Q&A. Unless you got some more comments to make on the paper. Um, any last minute thoughts on it? What when you read the paper, what was your initial reaction to it as far as that goes?
Well, I read the first, you know, the 17-page paper that came out in Nature, and I'm like, they're really not telling me much of anything here that I want to know. I mean, I want the bottom line compared, you know, chimp compared to human, and not really getting it. And so, in fact, it was actually kind of pro-evolutionary. It was really pushing the human chimp evolution agenda more than their 2018 paper, which was considerably more candid. Um, and I immediately saw that and thought, you know, the data has got to be in the supplemental material, and I know it's going to take some time to go through this 171 pages, but I've just got to do it. So I did. I put my, you know, nose to the grindstone and trudged through that and found what I needed. So
Right. And to let everybody know, I think I have the paper linked in the description box below. I think I have the Y chromosome paper as well linked in the description box below. So you guys can review that paper if y'all want to. It is, as Dr. Rob Carter noted to me, very boring. It's very drawn out. Um, but it does include some interesting information concerning differences. Um, as I understand it, most of this information can be found in the supplement material of the paper itself. Um, all right. So if you're ready, we'll go ahead and start open it up to a little Q&A. Um, haven't got a lot of questions here. I was hoping for a little bit more people to come into the chat. Um, I had some people to tell me that they were going to be here, and but I don't see them in the chat. So I'm going to go ahead and open up, get at least a couple questions in. Maybe some others will send in some more. All right, let's start with the first one here. All right, from Keeping Up. He's asking, can we explore the question at what point in great apes life cycle is it claimed that the fusion occur? He's talking about chromosome 2 fusion, because that begs the question of the population genetics required to speciate.
well, I don't think the evidence is there that it did occur. And then the claim is not that it happened in an ape, uh, that it happened in a human ape common ancestor. So, um, yeah, I don't see the evidence in humans that it even happened. So, and of course, the problem is if it did happen, if you did during meiosis and you get two chromosomes fuse head to head, end to end, which has never happened in nature with telomere sequence. It's happened with satellite sequence and telomere sequence and satellite DNA sequence. Um, so let's say it did happen, you'd have a problem, you know, with you'd have a cell come together with odd chromosome numbers that would be highly detrimental to that embryo even surviving. So, yeah, in a population thing, it'd be very hard for it to even happen to begin with, if it could. Um, but I think it's a mute issue because the data I'm looking at with the fusion sites in the middle of a functional gene, the cryptic centromere is in the middle of a functional protein coding gene, a very big one actually. It is a transmembrane spanning gene that looks like a cell receptor and is involved in cell signaling. So both the so-called genomic signatures of fusion are actually functional things inside the middle of genes, functional genes.
Right. Um, some evolutionists have argued that there are papers that have shown telomere to telomere fusions. If I'm understanding those papers correctly, they are um, kind of like it's kind of like arguing in a circle because the writers of the paper look at the supposed fusion site, they look at what this has in common, and then they go to these other genomes and they look for similarities to that, and they say, see there's a fusion site. Is that my understanding of what they're doing, or what?
I have never seen a paper showing a telomere to telomere end to end fusion occurring naturally in nature. Like I said, I've seen other types of fusions where the chromosomes break off in the middle in satellite sequence, um, or towards the ends maybe, but I've never seen a telomere to telomere fusion in nature. Now that does happen in cancer cell lines in petri dishes in the lab, immortalized cancer cell lines, but those are not normal healthy cells. Um, but I'd love to see a paper if that's in fact true. But my research, I have not seen one.
All right, let's move on. All right, this is from Lynn Mitzi. What do the evolutionists, say about apes today? Are they still evolving to humans? Exactly. Um, what is their belief concerning ape evolution? My understanding is that they think we're continuously evolving. Is that right?
Yeah, I don't know why. The ape genome, the chimpanzee genome, the gorilla genome, the orangutan genome, the human genome, they're not improving and getting better. They're accumulating mutations and getting more corrupt. And so, no, we're not evolving and getting better. Uh, there's genetic entropy going on. I'm sure I don't know if you've had John Sanford or anyone with his group on your show before, like Chris Roop. But yeah, no, we're not evolving and getting better. And you don't see chimps evolving into something else. Chimps are chimps. The same thing with
Right.
gorillas and orangutans. We don't see them evolving, and that's a fundamental question. Do we see these things evolving? No, we don't. And do we see genomes evolving and getting better and acquiring wonderful brand new information every day? No, we don't. We see mutations accumulating generation by generation.
Right. Um, we don't see uh, uh, animals or people continuously evolving. That, if I'm understanding correctly, evolutionists say that we're still evolving. But and a lot of times we ask, what are we evolving into exactly? So I mean,
Well, actually,
go ahead.
actually, you have a transhumanist movement that says we need to jumpstart human evolution. We need to begin merging man with machine. We need to begin doing genome editing. And so you have this entire transhumanist movement saying evolution is not moving along fast enough for us. We need to take things into our own hands. And so, yeah, you have that aspect of human evolution going on by a bunch of crazy, wicked people wanting to do all those things.
Yeah. I don't see any more questions. I guess we can spend the last few minutes just kind of wrapping things up. Uh, where do you see this topic matter going in the future? Do you wait a minute? I got a question. Yep, awesome. I was wondering when Donnie was going to show up. All right. Um, Question. It is said that using the same methodology that gets humans and chimps at 85%, we also get gorillas within themselves at 85%. He's talking about what critics have tried to argue. Did gorillas change by 15% since the flood?
I don't know. And I don't know about that 85%. I guess I'd have to go back and look at that paper. Um, I'm sure gorillas have changed somewhat since the flood, as far as maybe just diversifying within their kind. I know chimps have. We have bonobo chimps and then the regular chimps. And of course, they could still interbreed with each other, so they're not separate kinds. Um, yeah, I mean, there has been diversification within kinds since the flood. And so I definitely agree with that. Some creatures can diversify more than others, but they're still the same created kind.
I guess the idea is that they expected them to be I guess more similar to each other, and the fact that they diversified much more than expected. But the reality is that the God could created them with any number of a genetic amount of material, and then they diversified from that point. But they still stayed the same created kinds. That they didn't like diversify so much that they became outside their own kind. Um, of course, within our
Well, they can,
position,
yeah.
yeah, with our position, it's the types of differences that matter, and not the amount of differences or amount of similarity. So
Well,
we wouldn't look at. Yeah, go ahead.
yeah, I would like to say something along this line. So they've actually compared humans to humans. And so in the past, we're told based on single nucleotide polymorphisms or SNPs, that's a single nucleotide change between one human genome and another, based on SNPs, and they've sequenced thousands of human genomes all over the world. They have originally told us that any two humans were 99.9% similar to each other. Well, now that we have this new technology for long read DNA sequencing, now they're finding out that based on structural variance in between humans, any two humans can be up to 5% different from each other. And so based on structural variation between humans, between humans, uh, say between gorillas, between chimps, between orangutans, there is a significant amount of structural variation that can change that percent similarity. And then now we're
There is
discovering this with
go ahead.
yeah with this new with this new DNA sequencing technology where we can create 30,000 to 50,000 base reads. Now we can get through these areas and see what the structural variation really looks like.
Yes. Uh, Donnie has another question here. I'm gonna play that one. If animals within the same kind differ by 15%, why is a 15% difference between humans and chimps differentiating evidence for creation versus evolution debate? Um, I guess the idea is that critics are saying that if animals within the same kind differ by 15%, why is a 15% difference between humans and chimps differing evidence for creation versus evolution debate? Why would he's asking why would it matter as within our perspective? Why would it matter on the whole debate between creation versus evolution if there is a 15% difference between chimps and humans?
Well, I think I just answered that was talking about the structural variation. So that's why we see this big variation within a kind. Um, like I said, I've got two different studies that show I've got two different sets of data. I've got uh, I've got data that is very similar to chimpanzee, and then I've got data that is very dissimilar to chimpanzee. And then we've also got a completely other data set, which we don't have right now. What's present in human, absent in chimp, present in chimp, absent in human. So actually, we have 3 different types of data sets, really. And so you just can't say broad brush this 85% thing. You have to look at all these different data sets, what kind of genes are in those as far as long non-coding RNA genes, protein coding genes, that kind of thing. And so, yeah, and then with the structural variation within created kinds that's there, uh, you got a lot of different factors to look at. The fact of the matter is, we have
Um,
a large portion of our genome that I've shown in 2 of my studies that are very dissimilar to chimp.
right. I have a follow-up question on that. On uh, do you see the do you see that the differences are more that we should be focused more on the differences rather than the percent similarity? Because within the whole creation versus evolution controversy, um, uh, similarities can be accounted for by common design. And so, if you just look at the similarities, both sides claim similarities. So it becomes more productive to look at the differences because that's where evolutionists actually struggle on is trying to account for the differences between humans and chimps. Would you say that the differences be more productive to look at than just trying to look at the similarities themselves?
Oh yeah, definitely. I've actually got a semi-technical article coming out in our bi-monthly magazine called Acts and Fact, and it's going to be, I think, coming out this fall. And it's on the topic of orphan genes. So these are genes that are found in one. There's different levels of orphan genes, but let's talk about species-specific orphan genes. Uh, these are genes found in one kind of creature that are not found in any other creature, including those that are it's supposed to be related to evolutionary on an evolutionary scale. So, anyways, yeah, that's what we really need to be talking about is what are the differences and what's there, what's making them different. You can't just broad brush this with these percentages we're throwing around. So, anyways, I think that's important. So yeah, I've got a paper coming out kind of on a topic that's very closely related to that issue, coming out in a couple of months, I guess, month and a half or so.
Right. I actually have a question of my own on that as well. Uh, I have a question of my own on that. Uh, with the I'm trying to remember what my question was. The differences between humans and chimps. Uh, oh yeah, now I remember. Does evolutionists struggle? Would you say that this is a evolutionists believe that the split happened between humans and chimps, or happened between humans and whatever our allegedly ape-like ancestor is that they claim that the split happened between humans and chimps or humans and apes. Um, uh, that allegedly that happened about 67 million years ago. And then we know how slow mutation rates are. Do you see that this 15% difference would that be like a major blow? Because I mean I understand that from a worldview perspective, all they have to do is try to push their timeline back, but there hasn't been enough time within that alleged 6 to 7 million years ago for to have that much differences. Is that how you would look at it?
Well, I think I talked about this already in their statistical models, assuming a neutral model of evolution. That's how they basically look at this. That's highly hypothetical. You're calibrating your model with deep time, and you're only using regions of the genome that are highly similar, close to 99% similar for your model to work. There are limitations there. You can't use these regions that are highly dissimilar. Um, so, anyways, I'm not sure if I answered your question or not, but yeah, I think we've already
Okay.
kind of touched on that.
All right. Here's another question. I think it's from Donnie. Um, how do we explain species-specific orphan genes where one species of the same kind has a set of orphan genes and another doesn't? Does that mean orphan genes have been lost since the flood?
Well, we see these massive differences in orphan genes, particularly in the social insects like ants and bees, and different species of ants and bees. Uh, for example, like the leafcutter ant has got over 30,000 genes in it, and 1/3 of those are species specific to the leafcutter ant. And of course, I don't think the leafcutter ant can interbreed with other types of ants. Of course, the word species isn't really an accurate word all the time because there are species that can interbreed, which means they're the same kind, and there are species that can interbreed with other species, which means they're likely not. So, yeah, I don't know. That's how do we explain? Yeah, I don't know if you can lose orphan genes or not because many of these orphan genes are specific to specific types of anatomies and traits. When you get into behavioral traits, like with ants and the complex things that the ant colonies do, it's just off the charts, or the bees do in their colonies, and even when they go out foraging and to get food and come back and process it, and they all have all different kinds of casts, uh, of morphotypes of these behavioral insect species. Very complex situation. So you're going to have all these genes that are just specific to those behavior patterns, to those morphologies, to those caste systems that they develop within their colonies. And so that's why, compared to say a solitary insect like dipterans or flies, you have considerably more orphan genes in Hymenoptera, ants and bees and hornets than you do in flies. But it's a complex question. I don't think we can really get to the
Right.
bottom of that right now. Yeah. But anyways, I need to kind of get going here, so
All
let's
right.
we
I'm
could wrap
gonna
things up.
yeah. I'm. Is it okay if I show one more question? Donnie had
Yeah, then
another
we'll stop.
question. All right. If orphan genes can't be lost, why do orphan genes vary within species of the same kinds?
Well, we don't know there's species within the same kind, and I'd like to see an example of that. I guess um,
He does a
it's
follow up
it's
here.
it's yeah, I mean it's possible.
Example, both chimpanzees and bonobos have genes that are not found in the other.
I think they have some DNA sequence that's not found in the other. I don't know about genes specifically.
All right.
I mean, it's possible
Um,
you have genes that occur in gene families, and so even between dogs, then dogs are in the same kind. Wolves and the canines we have as pets can interbreed and are in the same kind. You definitely have variation in these amylase gene families, or odorant receptor gene families. There are variations between interbreeding species within that kind. Um, and so, yes, you do have that variability. I think it's just built-in genetic variability that God put into these creatures so they could diversify and adapt.
right. Um, all right. Thank you, Dr. Tompkins, for all your answers today. And I appreciate you coming on with us and discussing that paper. That was kind of like a big blow to the evolutionary community. I'm glad I actually been wanting to talk about that paper ever since finding out about it. Um, I actually discovered that paper completely by accident. Because I was looking for see if there was any papers that talk about the differences that didn't align in the between humans and chimps, and I came across that paper. I actually came across Luskin's article first, and then I came across that paper, and so that was pretty cool. But anyway, thank you again, Dr. Tompkins. We really do appreciate it. Um, and we'll I'll catch you later. Thank you again for all your answers. I know you live a very busy life, and so we do appreciate it.
All right. Thank you. God bless you. Thank you so much for what you do.
Thank you. God bless you too.
All right. All right. Bye bye.
Bye.